This is the recording on Chapter 4 with supplement on the hypothalamus and its hormones. Note that I've switched my pointer to the pencil here so that I can do a little bit of drawing on some of the first slides. I draw your attention to the fact that this is slide number 42 out of the lecture slide file named Chapter 3 and 4. And this will be a discussion on the hypothalamus, taken primarily from the endotext. The link is provided here, and it's also on the website, as you know. So we start first with this illustration. Actually, it's a photomicrograph of the midline of the human head. We notice, of course, the nose and the back of the head here, the cerebellum, the cerebrum, the brainstem, corpus callosum that connects one cerebral hemisphere to another. And I'll draw your attention to a few features of the hypothalamus before I draw the outline of the boundaries of the hypothalamus that can be seen on this slide. So first, of course, we recognize the pituitary gland sitting in its little bony cavity, the sella turcica, the sphenoid bone. We note the optic nerve would be coming from the eye to here to form the optic chiasm. And then below the corpus callosum, there's this structure. This is just off, that would be the left side, we'd be on the right side looking at it. It's just off to the right side, so we see this midline structure called the septum pellucidum. So we can't really see all of the thalamus, but there's a little bit of the the thalamus back here to guide us in our upcoming illustration. And then the thalamus with the hypothalamus here, of course, joins with the midbrain, and then the pons, and then the medulla oblongata of the brainstem. So now that we have that level of orientation, just one final one. This is the ventral surface of the brain with the pituitary stalk coming down here. So the pituitary stalk obviously connecting the pituitary to the base of the brain. I draw your attention again to the optic chiasm, where the two optic nerves come together, and one half goes to one side and one half goes to the other. Therefore, it's the chiasm or crossing. I'm going to draw a line from the optic chiasm to this white structure here. This is the anterior commissure. So if I can write it, I can't do it too well, but anyway, that's the anterior commissure. And this line drawn from the anterior commissure to the optic chiasm highlights the anterior boundary, or the rostral boundary, rostral toward the nose, of the hypothalamus. The structure that connects the optic chiasm to the anterior commissure runs along there, and that is called the lamina terminalis, so I'll put an LT here. So the lamina terminalis from the anterior commissure to the optic chiasm is the anterior boundary or rostral boundary of the hypothalamus. If we follow the base of the brain, which obviously is the floor of the hypothalamus, or the inferior boundary of the hypothalamus, back toward the brainstem, we see this bulging structure called the mammillary body. And when we draw a line from the caudal or posterior aspect of that, basically straight up to the thalamus, stopping just where there's a little depression there, that forms the posterior or caudal boundary of the hypothalamus. Then between the thalamus and the hypothalamus, there's a hypothalamic sulcus, which isn't really visible here. It may be a little straighter than my line. And that hypothalamic sulcus is the dorsal boundary of the hypothalamus. So if we could trace the lower margin, including the pituitary stalk there. I didn't quite get around the mammillary body quite so well. That is the hypothalamus. Seems quite small related to all the other size structures in the area, but those are the boundaries that can be seen from this view. We have the rostral and caudal, kind of imaginary lines, although the rostral does have a structure there, the lamina terminalis. The dorsal is kind of imaginary, but the hypothalamic sulcus is there. And then obviously the ventral boundary is the base surface of the brain around the pituitary stalk. Later we'll look at cross sections, and we'll look at the front, and we'll be able to see the lateral boundaries of the hypothalamus when we get to that point. So relatively small mass size compared to other areas of the brain that have very important functions, but many complex functional capabilities exist within the hypothalamus. It's really quite astounding, and you'll see that as we proceed. There comes the next slide. Very slowly. Come on, computer. There you are. So again, I'll use my pen function to outline the hypothalamus on this higher magnification. That's also showing some of the nuclei, collection of nerve cell bodies within the hypothalamus itself. In this case, again, we're looking from the right side, but now the section is deeper into the left side, so we're actually into the parenchyma of the hypothalamus itself. In the previous view, we were looking onto the surface of the hypothalamus, low mag, so we couldn't really tell. So again, the nose would be in this direction, so this would be rostral. And then this is toward the tail or the back of the body, or caudal. So anterior, posterior, dorsal, ventral, or rostral, caudal, dorsal, ventral. So here's the thalamus. And here's the fornix coming from the hippocampus, goes straight to the mammillary bodies. That was within the septum pellucidum that we saw. The S here is showing a little bit of the septum in that area. So with the thalamus here, we can draw our hypothalamic sulcus out here to the anterior commissure, and from the anterior commissure down to the optic tract. We're a little bit past the optic chiasm at this point, and then a line drawn from the hypothalamic sulcus to the point just caudal to the mammillary body. So there's our caudal, dorsal, and rostral boundaries of the hypothalamus, seen on this view. So again, this is the optic tract just after the optic chiasm here. Because of our plane of section being a little bit deeper, we're seeing the chiasm forming the optic tract. So several nuclei are present, are seen on this plane of section. First, above the optic nerve, we see the supraoptic nucleus. Then we see the preoptic nucleus. It's actually a little bit in front of the optic chiasm, so preoptic nucleus. The paraventricular nucleus of the hypothalamus, so just off on either side of the ventricle, right? So the lateral ventricle would be over there, coming down to the interventricular foramen into the third ventricle. So it's the third ventricle of this cerebroventricular system that we're referring to when we talk about paraventricular. As we move caudally, we see the dorsomedial nucleus. I may not be outlining their exact boundaries, just I'm outlining the letters. Then obviously below it would be the ventral medial nucleus of the hypothalamus. Posterior hypothalamus, hypothalamic area is seen here. And then they have an IF for the infundibular nucleus, which is actually the arcuate nucleus here. And we'll see that again as we proceed to get an appreciation for how it's an arching structure from the third ventricle farther out. If we can go into the plane, into the substance over there, it would arch out. And I guess we best wait until we're looking at a coronal section before we see that. And then, of course, the mammillary body here. So these are just a few of the hypothalamic nuclei. many of which will be important in neuroendocrine function that can be seen in this plane of section. However, deeper planes of section or other orientations of plane of section will show other nuclei. I don't want to leave you thinking that these are the only nuclei, of course, of the hypothalamus. So let's then proceed with our discussion describing the hypothalamus in the terms of zones, nuclei, and related functions. So those will come as we proceed. So this is a ventral view from the bottom of the human hypothalamus. We see the pituitary stalk right here in the center. And if we would want to, we could draw our boundaries. The anterior boundary here at the front of the optic chiasm. So we see the optic nerve coming in to form the optic chiasm, and the optic tracts go on back to the optic tectum of the midbrain. And from the optic tract back here to just to the side of the mammillary bodies, you can see them bulging there. So a line back here would be the caudal boundary. And then again on up there. So these are our boundaries of the hypothalamus from the inferior view. So the lateral boundary is a line from following the optic tract back to the lateral edge of the mammillary body. Now remember, we would have to split this down the center where the third ventricle would be. And since we're looking from below, this would be right and this would be left. There is a right and a left hypothalamus. It is a paired structure, symmetrical structure. The pineal gland is the only unpaired structure of the central nervous system. So then we can see the zones. There's a periventricular zone in red here, so right and a left. And in the yellow, the medial zone, and then the bluish color is the lateral zone. So there are three zones of the hypothalamus: periventricular, next to the third ventricle, medial zone, and then the lateral zone. The periventricular zone contained, as we'll see, the nuclei that we just described, most of which make up the endocrine hypothalamus. I'll just quickly point out other structures here. Here's the thalamus, the cerebral peduncle, major heavily myelinated pathway going from the cortex down to all the rest of the brainstem, and most importantly carrying motor axons down to the spinal cord to control the somatic motor system. Also note these anterior perforated substances and the posterior perforated substances, reminding us that there are many small vessels that enter into the brain tissue in this area, and obviously those coming to provide the vasculature to the hypothalamus and to the pituitary stalk, and forming the hypothalamo-pituitary portal system, vessel system, in this area as well. So there's the circle of Willis, if you're familiar with the arterial input to this area, that provides branches for all of these structures. So now I'm going to change my pointer to the arrow, and then have it I'm sorry, I can't read that. I'll try to keep the arrow active so it doesn't disappear. It's kind of frustrating. Okay, so let's read through this list of major hypothalamic cell groups based upon the zones. There's the periventricular zone, the medial zone, and the lateral zone, as we said. The nuclei within the periventricular zone include the arcuate nucleus, the periventricular nucleus, the paraventricular nucleus, and the suprachiasmatic nucleus that we won't have a chance to see. It has an indirect role in the biological rhythm of the neuroendocrine system, In other words, or more specifically, it regulates the rhythmic release of the glucocorticoids and melatonin and several others, and regulates the sleep-wake cycle. So it's a very small nucleus and is tremendously powerful in affecting the biology of the entire organism, sleep-wake cycles, corresponding endocrine cycles, and many of the biological rhythms. The medial zone, I got off on the function there a bit, but I wanted to talk about that now because we won't include it in the later discussions specifically for the endocrine function of the periventricular zone. In the medial zone, there's the anterior hypothalamic nucleus, which is right with the preoptic area that we mentioned before. So they're kind of one and the same, to be safe in general at the moment. The dorsomedial nucleus we saw, the medial preoptic nucleus, also out in the preoptic area, but closer to the midline, obviously, than the preoptic area within the anterior hypothalamic nucleus, and the ventromedial nucleus. I've got to try to... This pen color. Oh, I know I'm driving you crazy with this. I'm sorry. Okay, I think I got it that time. Sorry about that. Thank you for sticking with me. The lateral zone then, the lateral hypothalamic area or nucleus, the lateral preoptic nucleus, and the supraoptic nucleus are included. So you should be familiar with these. Let's follow the preoptic area, at least through the medial zone and the lateral zone. We see an anterior hypothalamic nucleus that contains preoptic area. Of course, it is in the rostralmost portion in front of the optic nerve and optic chiasm, obviously, in the medial zone. And then if we were in that area and moved medially, we would find the medial preoptic nucleus. And then if we went much farther laterally, we would find the lateral preoptic nucleus. So the naming of the preoptic nuclei is a prime example of this association with the zones. The medial more portion of the medial zone is where we find the medial preoptic nucleus. And then as we move laterally, there's this anterior hypothalamic nucleus area that is still preoptic in position and shares some functions. And then as we move farther laterally, the lateral preoptic nucleus. So even though no functions are listed for the lateral preoptic nucleus, you can kind of blend together anterior hypothalamic and medial preoptic functions with that. Obviously, there's still much more we need to learn about the hypothalamus and its functions. Thus, the arcuate nucleus then of the periventricular zone. Here we find the synthesis, neurons that synthesize TRH, CRH, GNRH, GHRH, and GHRH, growth hormone releasing hormone, and GNRH, used to be called LHRH. There are nerve terminals from here for the tonic release of the GNRH. So tonic levels of stimulation to the pituitary gonadotropes. Dopamine neurons here project into the primary capillary plexus from the tuberoinfundibular dopamine neurons, so opportunities for interactions among the systems regulating the HPG axis, the hypothalamo-pituitary gonadal axis. The periventricular nucleus, synthesis neurons that synthesize CRH, TRH, and somatostatin. The paraventricular nucleus, here we find oxytocin, vasopressin, and the neurophycins associated with each of those in the magnocellular neurons. As well, in the smaller neurons, obviously magnocellular, or the big neurons, they project to the posterior pituitary, dumping directly into the blood, so they have to be big neurons making large amounts of these hormones to produce significant systemic plasma concentrations. The parvocellular neurons that only have to synthesize enough to produce significant, physiologically significant plasma concentrations in the portal vessels to get to the anterior pituitary. The parv or parvocellular system both means small. These are the neurohypophysiotrophic neurons that are going to regulate the posterior neuro and anterior hypophysis pituitary. So the TRH and the CRH are found within smaller neurons that are distributed to this area, and we'll see those large and smaller cell locations in a subsequent slide. In the medial zone, the anterior hypothalamic nucleus with the preoptic area coordinates parasympathetic nervous system functions, temperature regulation, vasodilation in response to hyperthermia. So already we're beginning to see with the suprachiasmatic nucleus and the anterior hypothalamic nucleus functions that are not endocrine. Therefore, the hypothalamus is endocrine tissue. It is mixed function, endocrine and neuro-mediated functions. The dorsomedial nucleus, here we find neurons that synthesize TRH, CRH, and somatostatin, but it also has other neurons within circuits that regulates the autonomic nervous system activity and controls aggression. The medial preoptic nucleus, we find neurons synthesizing GnRH and TRH, and also these are associated with stimulating the GnRH and the prolactin surges. So we saw the tonic HPG function back in the arcuate nucleus of the periventricular zone, and the pulsatile function here in the medial preoptic nucleus. In the posterior hypothalamic nucleus, we have temperature regulation, responses to cold, regulation of the sympathetic nervous system and other visceral functions, regulates fight or flight response, so coordinating behavior with visceral organ function through the sympathetic nervous system, and influences sleep and arousal. The ventromedial nucleus, synthesis of GHRH, somatostatin, CRH, PRH, prolactin releasing factor, PRF, pardon me, I read the acronym wrong, TRH, it regulates insulin and glucagon secretion, controls digestive system functions, detects blood glucose levels, the glucoreceptors, and regulates food intake and the female sexual behavior. A new important function of the ventromedial nucleus is to detect an endocrine signal called leptin that is synthesized and secreted by fat cells. So that's a new area of investigation into a hypothalamic axis, the adipocyte or fat tissue hypothalamus axis. The lateral zone, the lateral hypothalamic area, is controlling hunger, regulation of sodium balance, and other types of functions. These are brief lists of the functions, and that's an important idea to keep in mind. The supraoptic nucleus, this contains magnocellular neurons for ADH, antidiuretic hormone, also called vasopressin, or we'll see it labeled as AVP, arginine vasopressin. There's also oxytocin and neurophycins from the magnocellular division. This is involved in the regulation of thirst and drinking and the synthesis of CRH in the parvocellular division. So again, this is an abbreviated list, but it's more than enough for us right now. I'm sure you will agree. So the next slide is a coronal section through the rat forebrain taken from the Paxinos and Watson stereotaxic atlas. It is at the level of the paraventricular nucleus of the hypothalamus, either abbreviated as PVN for paraventricular nucleus, but there's also a paraventricular nucleus of the thalamus, so I like to use PVH as the abbreviation, the paraventricular hypothalamic nucleus. Regardless, let's not be concerned about that. We note, as a stereotaxic atlas, there are numbers for dorsal-ventral and medial-lateral coordinates, but also the anterior-posterior rostral-caudal coordinates here measured from Bregma. I'll draw your attention to this. This is at the level of negative 0.94 millimeters. We'll see another level, the level of the arcuate nucleus or the median eminence in a moment, and I'll remind you of the distance that we've traveled from this level of the paraventricular nucleus to that level of the median eminence. Remember, rostral to this, we see the optic tracts here, so we are already caudal to the level of the optic chiasm, so there's a lot of hypothalamus in front of this level as well. I just like to use these two levels, the level of the paraventricular nucleus and the level of the median eminence, arcuate region, as examples to make some points. I'm not going to switch to my pen again, but you can start at this little sulcus at the lateralmost edge of the optic tract, and, whoops, I pushed on it as if it was my pen. Oh, come on, computer. Slow computer day. Draw a line directly dorsal. In this case, in the rat, the thalamus has a tremendous midline adhesion. It's not as massive in the human. It's much smaller. So the lateral ventricles open right into the third ventricle, which is bigger, top to bottom in the human. At least it's continuous than in the rat. But the top of the third ventricle that we see here would be where the hypothalamic sulcus would be. So we would draw a line up from the optic tract and across, and that would outline the entire hypothalamus, left and right. I guess we never talked about the midline boundary, the third ventricle, right? So that takes care of the boundaries on this. We notice that there are many little dashed outlines in these areas for the different subdivisions of various hypothalamic nuclei. We see a little bit of the anterior hypothalamic preoptic area still persisting in this area. The lateral hypothalamus can be seen here in different portions of the paraventricular nucleus, which is what we're going to look at more closely in the subsequent slide. So I focus on this one first to make the point again about the parvocellular and magnocellular components. The parvocellular controlling the anterior hypothalamus, the anterior pituitary, and the magnocellular neurons controlling, obviously, or releasing their hormones at the posterior pituitary. This is not quite 100%, but it is in the mid-90% range. There again, there's that risk of generalities versus details, but we don't need to worry about it on the organism level, at least clinically in humans. It's safe to say magnocellular is posterior and parvocellular is anterior pituitary function. So we have three levels in panel A, B, and C as we would proceed along that atlas. And again, this is the rat. However, the human paraventricular nucleus is organized roughly in the same fashion. At the anterior level here in panel A, This would be the anterior or the rostral level. And the third ventricle is here just off to the right. They're just showing the left hypothalamus, paraventricular nucleus in each case. Oh, I forgot to point out one thing on the previous slide. Notice the fornix here. It comes from the hippocampus, kind of a C-shaped structure. We saw it farther forward in the septum, and then it dives into the hypothalamus here and will continue all the way back, can be followed until it gets down to the mammillary bodies. So the fornix is an important landmark as well. And actually, if you draw an imaginary line through that, that would separate the lateral zone from the medial zone. And then there's not much of a landmark any place to differentiate the periventricular from the medial zone of the hypothalamus. But the fornix is an important landmark to separate lateral from medial hypothalamus. Let's not get lost there too much. I just want to draw your attention to the fornix because it's a key landmark that would be located off here, just lateral to the hypothalamic paraventricular nucleus. So there's an anterior parvicellular nucleus. And then as we get to the middle level of the paraventricular nucleus, again moving from front to back, rostral to caudal, we see at this level is the magnocellular nucleus or subnucleus, and then the parvicellular is divided into a dorsal parvicellular, medial parvicellular, ventral parvicellular, and then a periventricular parvicellular subdivision. We get into the caudal level or posterior level. There's a posterior magnocellular cell group with the periventricular parvicellular continuing and a lateral parvicellular subdivision as well. So obviously heavily studied, investigated using many different techniques to delineate these subdivisions of the hypothalamic paraventricular nucleus and all the other areas that we've talked about as well, and nuclei of the hypothalamus. So it's a very complex, compact structure, and it's difficult to dissect out all the different functions related to each thing, but we'll see how the neuroendocrine cells were identified in just a moment. So now we can go to the level of the hypothalamus, where we'd find the first parts of the median eminence before the pituitary stalk would be formed. We notice the fornix is now dipped deeper in. Eventually it'll come down, coming back out of the screen at us to the mammillary bodies, excuse me, posteriorly. So actually a line on the lateral part of that and the medial part of that separates medial hypothalamus from periventricular, and then lateral hypothalamus zone out here. So the optic tract is now moving up. It's going to go all the way up to the midbrain when we get there. So now we are drawing an imaginary line up from there, across the top of the ventricle, over to the optic tract on the other side, and down. So here we are seeing still the right and the left hypothalamus separated by the third ventricle. So using the fornix as our landmark, the medial zone of the the periventricular zone of the hypothalamus is here, with the dorsomedial nucleus and various subdivisions, a dorsal and a ventral and a central, and the ventromedial hypothalamic nucleus with a dorsomedial, central, and a ventrolateral subdivision. Part of the tuberal nuclei are coming in here, and then the lateral hypothalamus is becoming smaller. There's a perifornical region. So you can see these subdivisions and subnuclei become extensive as you get into the detailed study. Here on the right-hand side of the third ventricle, I point out the arcuate nucleus. So now finally we can see it arching from the third ventricle down to the base of the brain. And there's a dorsal and a lateral arcuate nucleus. So you're welcome to get into the details of studying all this and put your memorization cap on for sure. It becomes quite complex. This is not only for the hypothalamus. You can see various labels in all these other areas, so you can be kept quite busy learning the specific terminology to communicate exactly what part of the brain, what nucleus, and what subdivision you are discussing at any one point in time. It gets pretty complex. Anyway, we note we are at bregma minus 1.82 millimeters. The PVH level was at minus 0.94, so we're not quite a millimeter caudal to the prior level. So a lot of hypothalamus in between, in other words, and there's still a lot of hypothalamus caudal to this plane before we would see the pituitary stalk come off going down to the pituitary. But now let's proceed to the discussion of this slide that shows the distribution at the anterior, middle, and posterior levels of the hypothalamus within the range that we just looked at, the different levels of the paraventricular nucleus, and then at the level of the arcuate nucleus or median eminence. So these panels A through F are from the first level that we looked at, the PVH level, and the arcuate median eminence level are shown in panels G through I. So this slide is illustrating the results of a study to determine the neurons within the periventricular zone of the hypothalamus that project to the median eminence. To accomplish this, they injected a tracer that will be picked up by terminals and carried back to the cell bodies, a retrograde axonal tracer, into the external zone of the median eminence, where they were hoping just to hit the primary capillary bed to identify the hypothalamic neurons that regulate the anterior pituitary. But the median eminence isn't very big, as you can see from this slightly more caudal level, and some of the tracer got into the internal zone of the median eminence, where you would find the tubero-infundibular pathway, the axons passing through the median eminence on the inner layer of it, right into the posterior pituitary, forming the pituitary stalk into the neural lobe, of course. So it's difficult to demonstrate it specifically, but this shows it quite nicely. So here we see in panels A, B, and C the more rostral levels of the of the paraventricular nucleus, and we see the third ventricle separating the right and left hypothalami, and the paraventricular nucleus on each side, with the anterior parvocellular and some of the periventricular parvocellular subdivisions of the paraventricular nucleus shown here. And then for there's a gap between the anterior parvocellular portion when we start picking up the magnocellular and the more posteriorly located parvocellular cells, but the periventricular parvocellular cell groups are quite dense throughout all levels. Remember, this in particular was where we found the TRH producing neurons. So we're beginning to pick up the magnocellular division, medial magnocellular division, as we're progressing caudally. and this becomes very pronounced at this level here. So we're seeing this lateral ball of magnocellular neurons that are likely to be projecting to the posterior pituitary. So the dorsal parvocellular and medial parvocellular, ventral parvocellular neurons, and the periventricular parvocellular neurons, many of them, we can't say all of them, but many of them project to the median eminence and are involved in the regulation of the anterior pituitary. As we get to the posterior level of the hypothalamic periventricular nucleus, we notice that that lateral parvocellular subdivision is not involved. We see some of the medial parvocellular and periventricular parvocellular portions are still containing cells projecting to the median eminence, but the lateral parvocellular subdivision does not. The lateral parvocellular subdivision has projections down to the brainstem and the spinal cord, and are very important in coordinating somatic and visceral components related to the endocrine function of the periventricular nucleus. When we get to the arcuate median eminence level, we see the retrogradely filled neurohypophysiotrophic, excuse me, not neurohypophysiotrophic, hypophysiotrophic neurons of the hypothalamus. So this is where we would find growth hormone releasing hormone, and GnRH and dopamine neurons regulating the anterior pituitary at the highest density, but there also are some TRH and CRH neurons here as well. But mostly when you think of the arcuate nucleus, you think of the GHRH and GnRH neurons. The GnRH neurons here will be involved in the tonic release of the gonadotropins. So obviously when we study the HPG axis in greater detail later in the semester, we'll come back to these critical facts that you're studying now. So it's kind of boring when just the facts are being thrown at you and you're not trying to integrate it, but this is the stage we're at, the introduction to this information. So building blocks, building blocks. So this slide is showing us some of the interactions between the different zones of the hypothalamus, the periventricular hypothalamus, the medial hypothalamus, and the lateral hypothalamus. They don't always act independently. They do communicate with each other, and we see the lateral hypothalamus with projections to the medial hypothalamus and to the periventricular hypothalamus, with the connections focusing first on input into the hypothalamus, so local interaction between the zones. Also information coming in from the neocortex to the lateral hypothalamus and from the lateral, so reciprocal connectivity. The medial hypothalamus also having projections to the neocortex. Somatic sensory cortex is an example. And also the lateral hypothalamus to the motor nuclei, subcortical motor nuclei, the subthalamus, and the corpus striatum. So we're beginning to see that the lateral hypothalamus plays a very important role in receiving and integrating a lot of the somatic and visceral sensory input and motor output information and sharing that with the other zones of the hypothalamus. The reticular thalamus is a very important sensory structure, and it provides some sensory information to the periventricular hypothalamus. Remember, this is where most of our neuroendocrine neurons are located within the hypothalamus. So obviously the output to the pituitary gland is restricted to this periventricular hypothalamus. But also, as with the lateral parvocellular division, subdivision of the periventricular nucleus, there's output, and that's only one example, the output to the brainstem and the medullary, visceral, and somatic motor nuclei, so impacting blood pressure, respiratory rate, and a variety of things like that. The medial hypothalamus also projecting there, reciprocal connections between the medial hypothalamus and the brainstem visceral sensory nuclei, so visceral sensory information can come to the medial hypothalamus and then get to the periventricular hypothalamus through that local circuitry. Reciprocal connectivity between the medial hypothalamus and the limbic system and the non-reticular thalamus. Also, obviously, that portion of the thalamus having a lot of interconnectivity with the neocortex. Let's see what else we got here. Lateral hypothalamus also to the brainstem, medullary, visceral, and somatic motor nuclei. The limbic midbrain area. Whenever you hear limbic, you're thinking emotion. Psychogenic factors, emotion, visceral function, reward, all those kinds of things, giving direct input to the endocrine part of the hypothalamus as well as indirect to the lateral hypothalamus, and the lateral hypothalamus also having output to the optic tectum, substantia nigra, and the cerebellum, very important in motor function. If you can remember all that, that's great. Try to summarize it somehow in your mind. Understand that there's somatic and visceral sensory information that comes into the hypothalamus, and that is shared back with those areas and within the hypothalamus to bring together behavior, somatic and the somatic motor behavior, visceral motor behavior. So you've got somatic and visceral motor responses and endocrine responses, as we've talked about with this integrated functional system. So the little hypothalamus packs a powerful punch. So that was a schematic representation of the major neural pathways connecting the periventricular, medial, and lateral hypothalamic zones, subdivisions called here, with the rest of the brain. It's very much a summary. You'd have to read the literature endlessly to get all the details. Anyway, groups with identical colors are functionally linked. So that comes from the endotext. And you should read a little bit more about the afferent and efferent connections there to get a better appreciation of the specificity and complexity of this. So remember, our original discussions were all about external and internal environmental information coming in to the organism, somehow reaching the hypothalamus, and in coordination with many other areas of the nervous system, making some decision to respond to a change in the external or internal environment that coordinates somatic motor, visceral motor, sympathetic and parasympathetic nervous systems are what you think about when you hear a visceral motor system, or autonomic nervous system, and the endocrine responses. So the hypothalamus, as far as functions of the hypothalamic-pituitary axis, or in general, the collective axes into this overall axis, neuroglandular axis, if you want to think of it that way. The hypothalamus has many functions and is one of the major regulators of homeostasis. It controls the autonomic nervous system, acts with the limbic system to regulate emotional and behavioral patterns, regulates eating and drinking, controls body temperature, and regulates diurnal rhythms. So potent power punch from this little part of the brain. It also controls the pituitary gland secretions. So the hypothalamus exerts its effects on the pituitary gland in two different ways, as we saw. Posterior pituitary, magnocellular neurons. Anterior pituitary, the parvocellular neurons, or parvocellular neurons. The hypothalamus receives input from external and internal environment, as well as having its own receptors, for example, osmoreceptors for the antidiuretic hormone. It receives stimuli from the somatic and visceral sense organs. A couple of examples, one for the somatic. Both of these would be focusing on the posterior pituitary, impacting the magnocellular neurons. So the somatic, the mammary gland mechanoreceptors, bringing about the release of oxytocin. Visceral sense organs, say blood pressure from the medulla. If the blood pressure gets really high, then you have to determine whether that's due to a hypervolemic load or a water load, or if blood pressure drops and it's due to hypovolemia, then antidiuretic hormone would be very important, or the vasopressin. Vasopressin named for this hormone because it can constrict blood vessels. When you have low blood pressure, you want to constrict blood vessels to try to bring blood pressure back up. If you have low blood pressure, it might be due to hypovolemia. So you want the antidiuretic effect of this hormone from the posterior pituitary to increase water reabsorption and keep the volume of blood you have. So this hormone will shrink the container, vasopressin, and reserve the volume in that container to try to maintain blood pressure. If blood pressure is too high and there's too much volume, then you decrease the ADH secretion from the posterior pituitary and allow the elimination of volume and the expansion of the container. So hopefully you could follow that explanation. But anyway, we'll build on those as we go through our studies. So these inputs travel via the medulla oblongata, coming up from the spinal cord, or other nearby sensory input through the cranial nerve nuclei. The vagus nerve is a prime example that has direct input to the medulla oblongata, and reach the hypothalamus through innervation of fibers producing dopamine, adrenaline, noradrenaline, serotonin, and acetylcholine. So these we will talk about in the next section when we talk about neurotransmitters, as well as fibers releasing neuropeptides such as enkephalins, neuropeptide Y or NPY, neurotensin, dynorphins, and endorphins. So all of these types of neurotransmitters, as well as many others, impacting the neurons of the hypothalamus, in particular, for our case, the neuroendocrine cells of the hypothalamus. And those neuroendocrine cells have to integrate all these types of neurotransmitter input to determine at any one point in time how much of their particular hypothalamic hormone they're going to synthesize and secrete. So it's very complex. The release of hormones from the pituitary is therefore subject to many different stimuli from higher centers acting on the hypothalamus. So you simply can stress yourself out from the level of the cortex and the limbic system, and that will increase the firing rate of the CRH neurons in the hypothalamus. A stress response, a psychogenic stress response, increasing the activity of the hypothalamo-pituitary adrenal axis. In response, or you can be scared, frightened, fear is a potent stimulus as well that can be generated from an external environmental input. So a Tyrannosaurus Rex is running at you, you see it, that gets somehow to the hypothalamus to turn on your HPA axis, right? As well as coordinate your somatic motor response and your visceral motor response, get your blood pressure up, get your respiratory rate up, and get your butt out of there. Excuse me. In response to stimuli such as stress, pain, and emotions, the hypothalamus can exert effects on the anterior and posterior pituitary gland in order to respond rapidly to environmental change, as well as to feedback from internal systems. So this makes a very important point about the feedback to allow things to be pushed away from baseline homeostatic levels, but also determine when appropriately to come back to homeostatic baseline levels. I want to draw your attention to the wording here. In order to excuse me, in order to respond rapidly to environmental change. So the neuroendocrine response might be rapid, and your plasma cortisol levels might increase very rapidly. But the effect of the plasma cortisol is going to be more delayed. So by now you should have looked at the HPA axis lecture and streaming video, and you understand the delayed action of the hypothalamic pituitary adrenal axis, hormone 3, the glucocorticoids, to bring about a delayed increase in plasma glucose and anti-inflammatory effects to help you recover if you escape from the T-Rex, right? Or it may just make you a sweeter tidbit for the T-Rex if you don't if you don't escape and discover a new survival strategy. Anyway, try to put it into place. So the hypothalamus is the least well understood area of the should be an H there, the hypothalamic pituitary axis, and research into certain aspects of its function is ongoing. So this is our progression: textbook, review literature, then primary literature as we proceed through the semester, trying to catch up with the contemporary research. An example is current work involving the recently discovered peptide hormone, leptin, that I told you about, that somehow gets through the blood-brain barrier and into the ventromedial hypothalamic nucleus, binds to its receptor, and has potent effects on the organism. So it has shed light on the existence of an adipose tissue brain endocrine axis, as I had mentioned. The complexities of the hypothalamo-pituitary interactions are the final discussion that we will have, and you are assigned a reading under the course content for this, a couple of articles, so you should check your emails to make sure that you get to those assigned readings and understand them, and the figures within those articles, most of them are review articles or review type articles, so that you are prepared for questions on that material for test 1. So the hypothalamic hormones are not always a one-to-one relationship with pituitary hormones. In other words, there's multiple secretogogues for any one pituitary hormone. There's crosstalk in the ultrashort loop negative feedback, might be through retrograde transport up the axon terminals. We saw the marker used to identify the retrograde transport, track tracing substance in that earlier slide. Well, it's thought that maybe, anyway, ultrashort loop negative feedback might do some signaling through that mechanism. It's pretty complex and theoretical, so let's not worry about it. Or maybe through the cerebrospinal fluid and ultimately get to the hypothalamic neurons so that one hypothalamic hormone can impact neurons of another hypothalamic hormone. Extrahypothalamic release of neurohormones. Well, this will be through neuronal circuitry. And when you're talking about extrahypothalamic projections of neurons, say a CRH neuron in the lateral parvocellular subdivision of the hypothalamic paraventricular nucleus, projecting to the brainstem, and then being released at a synapse onto another neuron there in the brainstem somewhere, maybe the medulla oblongata, you would refer to the CRH as a neuropeptide, not as a neurohormone. If it is acting in a neurocrine fashion, you call it a neuropeptide. If it is acting as a neurohormone, then you call it a neurohormone. So it just depends upon where it's being released, what term you would use for it. Thus, we proceed to neurotransmitters and then neuropeptides in this course. There's crosstalk among neuroregulatory inputs, the neurotransmitters. For example, glutamate and GABA. Glutamate is going to be the gas pedal, GABA is going to be the brake. And then most of the other neurotransmitters are going to be, or neurochemicals are going to be neuromodulators, modulating how much gas and gas pedal and brake pedal are applied to the neurons, the neuroendocrine cells. There's crosstalk among hormones 3. For those systems that have hormones 3, for example, the estrogens, and the pituitary endocrine cells of another axis, and we'll see some examples for that, such as the hypothalamic pituitary breast interacting on this slide with the hypothalamic pituitary gonadal system, and then on the subsequent slides, the hypothalamic pituitary axis interacting with the hypothalamic pituitary gonadal axis. Hypothalamic pituitary adrenal axis interacting with the hypothalamic pituitary gonadal axis, or HPA-HPG interactions. So this slide is showing us an example of the prolactin effect on gonadotrophin or gonadotropin secretion. So we focus first on a situation where there would be hyperprolactinemia in the systemic circulation. This is usually produced by a prolactinoma, which would be a type of pituitary adenoma affecting the lactotropes and therefore increasing, it would be a tumor of the cells producing prolactin, so prolactinoma. Adenoma means glandular tumor. So hyperprolactinemia is going to impact dopamine neurons of the arcuate nucleus, and because that's the negative feedback, right? When prolactin goes up, you have to increase the release of dopamine so that it would decrease the release of prolactin, synthesis and secretion of prolactin from the lactotropes of the anterior pituitary. But here we're talking about interactions, so the dopamine secretion that would be increased by the high prolactin would affect the GnRH release, inhibiting it, decreasing the gas pedal drive of GnRH on the pituitary gonadotropes, thereby decreasing LH and producing a hypofunctional gonadal situation. So that's one example of interaction, one of many. This slide is taken from one of your assigned readings, the HPA-HPG interactions between stress, HPA system, and the female reproductive system, the HPG system. It is an article in the Annals of Internal Medicine by Croesus et al., published in 1998, but still a very good review article. Remember, we're going from textbook to review article to primary literature, so let's get geared up. So in this table, we're seeing the stress system, which is the hypothalamic pituitary adrenal axis, or the HPA axis. The CRH, corticotropin-releasing hormone, inhibits gonadotropin-releasing hormone secretion, decreases GnRH release. Beta endorphin, which is produced, well, here they're talking about neurons within the brain, within the hypothalamus, producing beta endorphin that are activated by stress, inhibit GnRH secretion. Cortisol, hormone 3 of the HPA axis, inhibits GnRH secretion, inhibits luteinizing hormone secretion, and inhibits estradiol and progesterone biosynthesis, that would be peripherally, and also indirectly through these effects. And also, cortisol intracellularly in the target cells inhibits estradiol actions. The locus ceruleus is a major source of norepinephrine. We'll study it in more detail when we talk about the neurotransmitters. It is a brainstem nucleus. It's a locus, a location where the neurons look blue because they're making norepinephrine. So ceruleus means blue and locus means spot, a blue spot, right under the cerebellum and the pons. And this locus ceruleus, this center, is very sensitive to the external environment, any changes, and immediately will notify the more rostral areas like the hypothalamus, but also cortex and limbic system, of any changes in the external environment about which the higher centers should be aware. So that norepinephrine stimulates the gonadotropin-releasing hormone secretion. So here you have these overpowering effects that inhibit GnRH, whereas the norepi system is trying to keep it going a little bit so you don't completely shut down the HPG axis. You have the reproductive system. The HPG axis, estradiol stimulates CRH synthesis, so contributing to a vicious cycle up here when stress is excessive. Estradiol stimulates cortisol-binding globulin secretion, so you can transport the higher level of plasma cortisol during the excessive stress situation. Estradiol potentiates norepinephrine actions. So there's only one example of the norepi effect given here. Norepi will also, under certain conditions, drive the CRH neurons to synthesize and secrete CRH. Well, this slide is showing us the interactions of the reproductive system with the hypothalamic pituitary adrenal system axis and locus ceruleus, or the norepinephrine system, so we get to see more of what the locus ceruleus, or LCNE, does. So here's the stimulus. Here's represents the response, all these solid arrows, and these broken arrows represent the regulation, the feedback effects, and then the final effect. So we have stimulus, response, regulation, and the organismal effect within this example system. So stress is going to activate the CRH neurons that also contain vasopressin within the parvocellular portion of the periventricular nucleus of the hypothalamus, and then that controls ACTH release, synthesis and secretion by the corticotropes in the anterior pituitary, and then targets the adrenal cortex for the glucocorticoid synthesis and secretion. So here's the HPA axis. And then here are components of the HPG axis for the interaction. Now this is just showing the locus ceruleus contributing here. So stress activates CRH, activates CRH neurons to increase the release, increase ACTH, increase glucocorticoids. You get the negative feedback at the level of the pituitary and at the level of the hypothalamus, and then potential effects on the target tissues. Stress is also going to activate the locus ceruleus neurons. You'll have interactions between these two, the CRH and the locus ceruleus norepinephrine neurons. The CRH neurons are also going to drive the POMC, pro-opiomelanocortin, beta endorphin, and alpha MSH. Here they're representing both pituitary and neuropeptide aspects of this, but let's just leave it in a circle for now. That can also have an effect on the locus ceruleus. Anyway, let's follow the norepinephrine. The norepinephrine has the effect on GnRH, and the CRH has its effect on the GnRH, in effect decreasing the GnRH release and decreasing LH and FSH, decreasing the hormones 3. But whatever hormone 3 there is, it can have a positive effect on the CRH, basically to sustain this stress cycle and end up with elevated activation of the HPA and suppressed activation of the HPG. So if you think of the two basic functions of an organism, survival and reproduction, HPA and HPG axes to reduce it or minimize it as much as you could, I guess. under dire stress, chronic, extensive, stressful situations, external or internal environment, you will sustain the activity of the HPA axis for hopefully the good parts of the glucocorticoids. If it goes too far, it's pathological. And you're going to shut down reproduction. At times of severe environmental stress, it's not the best time for an organism to try to reproduce. In this slide, we can just, the last slide of this presentation, we can just quickly talk about the interactions of the the HPA axis and the HPT axis for the thyroid. So the HPT axis, there's the TRH neurons, TSH in the anterior pituitary, and then T3 and T4, the hormones 3. And then the HPA as we had discussed. So the red are going to be activation, and these blue lines are going to be inhibition, so negative feedback shown here. So hormone 3, the cortisol, is going to have a blunting effect on the thyrotropes at the level of the anterior pituitary, and so under stressful situations, you'll have a blunting of the HPT axis. So this schematic representation of the main factors interacting in the regulation of TSH synthesis and secretion. There's dopamine, somatostatin, alpha adrenergic pathways, noradrenaline. So the solid arrows, the red arrows were the stimulation, and the open arrows were the inhibition. So cold is a very potent stimulus through the noradrenergic system. Some of it from the locus coeruleus, but other areas as well as dopamine. So the adrenergic system is going to drive TRH, the hormone 1. Dopamine is going to drive somatostatin, and the somatostatin and the dopamine will blunt or have an inhibitory modulatory effect on the TSH, the anterior pituitary. Dopamine will also suppress prolactin, counteracting the effect of the TRH on the release of the prolactin. So TSH is an alternate or an additional secretogogue to prolactin release. It's not known what role this plays overall in the HPT axis functioning, but anyway. Then, of course, you've got the hormone 1 driving hormone 2, driving hormones 3, and the negative feedback effects. You also have neuroimmunomodulation if there is illness. The tumor necrosis factor alpha and 2 of the interleukins, the 1 beta and interleukin 6, can also have inhibitory effects on the thyrotropes in the anterior pituitary. So the HPT axis regulation is very complex, and as you know, there's probably going to be a more immediate and severe clinical pathology with a sustained disruption of the HPT, whereas sustained disruption of the HPA might take a bit longer to present clinically. Well, this, I give the source of this figure here below. This concludes our discussion of the hypothalamus. I hope you have enjoyed it too much and are not too overwhelmed by the level of detail. Continue to study and learn, and with time, all of this will become easier for you. I appreciate your